The overall goal of this procedure is to induce EAE consistently and easily in any mouse strain, including those reputed to be resistant to EAE induction. This is accomplished by first immunizing donor mice with myelin-specific proteins or peptide antigens emulsified in complete Freund's adjuvant. 10 days later, the lymph nodes from the donor mice are isolated and lymph node cells are re-stimulated in vitro by culturing them with the same antigen for 4 to 5 days. The cells are then adoptively transferred into naive recipient mice. Susceptible mouse strains will develop EAE in 8 to 14 days. To trigger EAE in a resistant strain, 14 days after the adoptive transfer, the recipient mice were then immunized with antigen in CFA. Using this method, EAE can be induced in resistant mouse strains.
The main advantage of the adoptive transfer and challenge protocol for inducing EAE is that it allows for reliable disease induction. Rarely do we see less than 100% clinical disease. We can tailor the protocol for inducing disease in susceptible mouse strains, but more importantly, we can use this protocol to induce EAE in many mouse strains previously considered resistant to disease. Demonstrating the procedures today is our lead research associate, Xiao-qing.
Begin by preparing a 2 mg per milliliter solution of MBP in PBS. Use a ground glass syringe equipped with a Luer lock to draw up 100 microliters of antigen solution per mouse to be immunized. Using a separate syringe, collect an equal volume of complete Freund's adjuvant H37RA. Then, with a 3-way stopcock, connect the 2 syringes,